I'm Gert van Capelle. I'm part of the Optical Imaging Center in the Erasmus MC in Rotterdam, the Netherlands.
And this is Tom Skillman from Immersive Science. This presentation provides an overview of some of the key uses of ConfocalVR in looking at complex 3D images.
The collaboration part is quite important, even within one university. Just the ability to work with more people and look at the same image, point at things, move them
Right,
around
right.
is very, very useful. These are U2 OS cells, so they are liver tumor cells. I load here the Heugt channel, so that's the blue channel, so I'm changing the color to blue and reduce the noise a bit. And there we clearly see the interface nuclei. Here, this is probably an anaphase, so these cells just divided, and here we have a prophase. So I go to the next channel, that's propylium iodide. So that's binding the DNA and the RNA. So you see it both in the nucleus and in the cytoplasm. I also reduce noise a bit. And you nicely see the nucleoli that are these spots here in the nucleus. So, and then the last channel, that's the membrane channel, PKH. So that's binding to the membrane. It's green. And in this cell, you very nicely see all these membranes. These cells are coming up when they are dividing, but they keep attaching to the glass. And that's what you very, very nicely see in 3D. That's very, very hard to see in 2D. And if we now put all the colors together, then this is the complete image now. So what we did here is we put a label in the DNA, and on that label we bind a fluorescent tag. And in this way, we can really follow the spot of the DNA in time. However, what we found out that if you do this, there is much more movement in the DNA than we expected. And here in 3D, you can nicely follow the spot all the time because you see all dimensions. You see X, Y, and Z. So this is more or less the only way in which we can very easily, very quickly see what happens with spots. Where are they going to? How long are they there?
Yeah, I'm just going to make the image a little bigger for a second to see. Yeah, so to our eye in VR, picking up this spot here and tracking it is really quite easy.
Yeah. So this is a human embryo. And what we see here is the cells that are going to form the epiblast. And that's the part these people are studying. So they are studying what's going wrong with the embryonic development in humans.
Okay.
And they want to find out what is going wrong there. And one of the things they want to know is how many cells are here in these clumps.
Okay.
So the very difficult thing is that we have a lot of noise in the background that is now nicely filtered out. And then we need to count the number of cells that are here. But,
and I was, if you don't mind, I was just going to show this background noise that you talked about. So if we grab the excluder here and just pull it out of the way and then change the invert, this is, and I'm going to make it a little bit smaller. Oh, too much going on here. Make this a little smaller just so we can see, but there's a lot of noise to deal with.
We did try a few softwares to do an automatic counting, both in 2D and 3D, but these cells are so hard to separate from each other that was really impossible. This is
Go ahead.
cells transfected with H2B and maple. And maple is a fluorescent dye that you can switch from green to red. So what we did do here is we switched a few chromosomes from green to red. And then we make a long time-lapse experiment just to see how these chromosomes end up in the daughter cells. Now here we do not have the whole time series, but what I wanted to show you that you can nicely see how the chromosomes are going to the equatorial plate, because that's a process that is hard to see in 2D, but here you can really see it happening in 3D. So if we play the movie, are you ready?
Yep.
Then you see these chromosomes nicely going to an equatorial plate. And it's coming in the other direction than you would expect, I should think. That would be the equatorial plate. But you see all the chromosomes nicely going in. And this is fantastic that you really see these individual chromosomes nicely lining up on the equatorial plate. Switch this color in. And we can either do this in an interface nucleus, but then you are not sure which chromosomes you are going to hit. So if you do it in a prophase nucleus where you already get condensation of the DNA, you can really select the chromosomes that you want to switch.